You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. My guest is Erbil Abaci, Assistant Professor at Columbia University Medical Center. We're going to be talking about tissue engineering of human hair follicles using biomimetic, meaning mimicking biology, approaches. So Erbil, thanks for coming. How are you doing?
Good. Thank you, Richard. Thanks for inviting me, and I'm very excited to be here on this podcast today with you.
Yeah, I've heard of people wanting to engineer skin, 3D print skin, grow skin for burn victims, etc. But why a hair follicle? Why is that so important to skin itself?
Well, skin is actually a very complex organ. It is composed of many different components. When you think of skin, it's not only the epidermis and the dermis. It has vasculature, for example. It has pigmentation. It has innervation, for example, sensory neurons, and it has this mini organ called hair follicle. It's actually a very complex organ by itself. And hair follicle is also a functional organ that talks to other tissues in the skin. It talks to, it communicates with the blood vessels, for example. It can induce the formation of blood vessels. It can, it talks to other types of skin tissues, such as the epidermis and all the other sensory neurons, for example. There is some sort of communication between these two different components of the skin. So hair follicle is obviously also a very important for aesthetic reasons. People lose their hair sometimes because of androgenic alopecia, this is male pattern baldness, or it can be alopecia areata, which is an autoimmune disease that is causing these hair follicles to shed in both females and males. So those are very especially for young people, those are very devastating diseases to address. Or in burn patients, for example, when you have a third-degree burn on your scalp, for example, versus on your some other side of your body which doesn't have any hair follicles on it, you want to restore it. You want to replace it with engineered skin that has all these functions and components to it, so that you can have a fully formed functional skin that matches also all the other properties of the site where you have the burn on.
So does the hair follicle act as a transport mechanism of material in and out of the skin, or what are its different functions that people have figured out so far?
Well, my research is actually looking into more of engineering of the hair follicle. I'm actually an engineer by training, and I got interested in the skin 4 years ago when I joined Dr. Angela Cristiano's group at Columbia University. She is a world-renowned expert in hair research, and I learned so much from her and in the lab at Columbia. From the dermatologists, hair researchers and skin researchers. I'm not a hair biologist, so I'm not the best person to talk about hair biology. But so from literature, we know that it signals, for example, these specialized cell type of the hair follicle, which is called dermal papilla cells. This is a mesenchymal cell type that is embedded in the dermal compartment of our skin, and that is close to the hair bulb, and it directly interacts with the epidermal cells overlaying it. And we know now that the keratinocytes, the epidermal cells, as well as these dermal papilla cells, they can signal through for example, they can signal vascular endothelial growth factors and other important growth factors that can induce blood vessel formation, because this is highly important for the regeneration of this tissue. We have hair cycle going on throughout our lifetime, and this endless cycle has different phases. And in the anagen phase, which is the growth phase of the hair follicle, this process requires newly formed blood vessels and a constant supply of blood to the tissue to have enough nutrients to complete this regeneration properly. So for this regeneration purpose, dermal papilla cells and keratinocytes, they signal, they basically secrete proteins into the dermis and attract basically blood vessels that are embedded in the dermis of our skin.
So they attract their own blood vessels. I mean, hair grows in existing skin. So you're saying that as it grows, and in order to grow, it's attracting vascularization to itself continuously?
Correct, yeah.
Okay, well, that's good, because I know that's a tough problem with a lot of tissue engineering, is getting adequate vascularization. So if you can understand that process, perhaps that can be used for other tissues as well.
Exactly.
So what makes the hair follicle so complicated? What is it about it that is tricky to make?
Yeah, it is super complicated. It's a very complex organ. It is composed of, when you look at it closely, it's composed of at least 8 distinct cellular layers. Of these these are epidermal layers, and they're specialized in for different functions, and they have to be generated at the right space, at the right time. So this is what makes it very difficult to generate. Also, to achieve this specific differentiation, you have to have certain interaction between these epidermal cells and the underlying dermal papilla cells I just told you about. So that's how hair formation occurs in the body. You have these aggregates of dermal papilla cells in the dermal compartment, and then on top of that you have the keratinocytes. Then these two cell types start to talk to each other, and then this makes them grow down into the dermis, so it extends. And while it is extending, also these keratinocytes are differentiating into these specific distinct cellular layers to form the hair, a complete hair follicle. And when you look at the hair follicle, you can also see these little pockets, attached to it. For example, there's sebaceous glands. They are the fat factory of the hair follicle. They're super important for the hair follicle. It gives the greasy function to it. And then there are the stem cell compartments, for example, in the hair follicle, because it's a constantly regenerating organ. So you have to have specialized compartments for stem cells in certain locations of the hair follicle. So it's very complex and very actually difficult to engineer. And the main challenge is actually, when these cells, especially dermal papilla cells, when they are taken out of the body and put in culture to expand, and later to put back and together with the other cells and engineer the hair follicle, when you start culturing these cells on plastic dishes, they lose their genetic signature. So they lose their function almost completely. So one challenge is to restore this function on these multiplied, expanded cells first, so that when you put them together with the other cells, they can start the physiological interactions and start forming hair. So that was one of the challenges that we encountered. And we use a genetic approach to reprogram the cells by overexpressing some genes, including LEF1, in these cultured dermal papillae cells, which gave them the ability of hair induction. And then we use these genetically manipulated, if you will, cells to engineer our hair follicles using 3D printing.
So what are some of the specific roadblocks in engineering the hair follicle? Like, what have you been able to do, and what still is a challenge to you?
Well, there's still so many remaining challenges. What we've shown in the paper is, so we've been able to use 3D printing to achieve the right conformation and arrangement of cells to grow hair, and then we take these constructs and we either leave them in vitro completely in the lab, without putting them on animals. We were able to grow human hair in vitro without any grafting. And then we tried to also graft it onto mouse, and we were able to grow human hair efficiently on mouse in these constructs. But the challenge here is when you compare the efficiency of hair follicles you get when you graft them onto mouse versus the efficiency of the hair follicle you get in vitro, is completely different. It's much higher in vivo when they're put on animals. And this is not surprising. It's not only specific to hair follicle, hair engineering. It's a general thing that we don't fully understand why we can grow better tissues in the graft when they're grafted onto animals versus when we keep them in vitro. And we don't know exactly the formulation, the chemical and physical signaling that happens in the body. If we figure this out and if we can recreate this signaling in vitro, we'll be able to grow as efficiently hair follicles in vitro. And this is, as an engineer, for me, this is real tissue engineering. For clinical applications, of course, you will have to put it onto patients, and it will work much better. It is good, which is great. But then to be able to engineer a tissue completely without exposing it to any body or any microenvironment, that is the dream for me in this line of research.
Well, cells communicate with each other, so there's this cell-to-cell communication that's instructing and giving feedback to the follicle on what to do. That's probably why in the dish it doesn't grow properly.
Yeah. Plus, we in the media, where what we use to culture the cell in this study, we only use a plain medium, which means that we don't have any growth factors or any soluble factors in it to induce, to enhance the efficiency of this hair growth. As opposed to, in mice, there are all these growth factors coming from the surrounding tissue, in the other components that I told you about in the skin, or even from other organs. We know that organs in the body, they communicate with each other. So they're communicating through blood vessels, through blood, and, there may be a signaling that we are missing that is coming from, liver, for example, that is released into the bloodstream and then that goes to the skin and induces hair follicle formation. So now we're starting to add all these important soluble factors and growth factors into our media to achieve the same with the same efficiency in vitro.
Well, does this work on mice? So you immunosuppressing the mice and then grafting on stuff to make you grow like hair follicles and skin, or how's it working with mice?
Yeah, we did it on mouse, correct, in immunodeficient mice. But we did it also in vitro. I'm just talking about the efficiency. We were able to grow human hair with fully formed human hair with a hair shaft in vitro for the first time. Actually, when I saw the first time growing, a hair fiber coming out of our constructs, I was running in the lab and yelling to people, first hair, it's human hair, and people came around and take pictures. It was amazing to me to see even one hair growing out of our constructs. But now I want to imagine the day where we would see hundreds of hairs coming out of our constructs, like it does in mice.
Well, in terms of like the vascularization, what's the size of one hair follicle versus the amount of vascularization it needs? From what I've heard in other tissues, you get more than, I don't even know if it's as much as a millimeter, but a very small distance away without a blood supply, the tissue dies. So what's the size of a hair follicle relative to that?
Yeah, that's a very good question. I don't have a clear answer to how many, like what would be the amount of blood vessel you would need to supply enough blood to a certain number or like certain thickness of hair follicle or certain length of the hair follicle with certain number of cells. I don't have an uh, certain answer to that, but it is very important. So what we did in the study was, first we tried it because first we didn't have any vasculature in our hair-bearing construct. So directly we grafted it onto mouse, and it didn't grow any hair. So and instead, when we looked at the graft, we saw necrosis. So the tissue was dying. Basically, our graft was dying within a week, and it's not long enough for hair follicles to grow. So we said maybe we can vascularize this. This needs blood vasculature as well. So using our previously established vascularization methods, we also added engineered blood vessels into these hair-bearing constructs. And when we repeated the same experiment, when we grafted them onto mouse, then our tissue remained alive long enough, and it gave our hair follicles enough time to grow and form fully formed hair follicles. So it's definitely very important to have this vasculature. The amount of vasculature, we didn't really look at the extent and extent of these vascular networks. We basically seeded endothelial cells. We embedded them into the dermal compartment, and we induced them to spontaneously form blood vessels and blood networks. And then what we saw after we grafted them on these animals, these engineered human blood vessels, they combined, they merged with the host vessels. So basically, mouse blood was running through our engineered blood vessels, which was supplying nutrients to the skin for their survival. But again, they were not hair follicle. They were not blood vessels around the hair follicles. They were randomly distributed in the dermis. So if you can find a way to get them closer to the hair follicle in the right amount, or in a sufficient amount at least, you may have this ability to regenerate and have the hair follicles undergo hair cycle higher numbers of times.
Oh, wait a minute. You said the blood vessels are randomly distributed. And what about mapping the hairs? What about looking at a sample of skin and mapping the hairs and then mapping the blood vessels and seeing there's a correlation? It probably must be. It can't just be random and the hairs are there and some vessels are far from hairs or some are close. And there's got to be at least some proximity in order for them to grow.
Yeah, that's something definitely that we can look into. And I'm sure we'll see if you see any, if you see them closer to the hair follicles, I'm sure you'll see more hair cycling there and a more survival of the cells and more dividing cells and better reservation of the stem cells of the hair follicle. And we don't know yet how many cycles our hair follicles can undergo, these engineered hair follicles. We have to monitor them long enough because human hair cycle is very long. It's not like mouse hair cycle, which is only 4 weeks. But human hair follicle hair cycle is it takes years. So it takes years for us to figure that out, how long it goes.
Hold on 1 second. So, question about your constructs. What is it constructed of? What does it look like, your construct so far?
So our constructs are composed of collagen type 1 gel, which is a hydrogel, and which is the most abundant extracellular matrix of the dermis. These are coming from rat tail, and uh, we have dermal fibroblasts embedded into these 3D hydrogel, and in this hydrogel we have our micro wells, or you can think of them as micro holes, which we made by using 3D printing. And in those holes, we add our keratinocytes and dermal papilla cells, and then they settle down into this channel. When they settle down, dermal papilla cells settle down first because we see them first. They go in, they sink into the channel, where they form these spontaneous aggregates. And when they form the aggregates, actually, they restore their hair inductive properties partially. And when they do this, after they do this, we add our keratinocytes on top, and they start communicating with each other. And that's how we form our constructs, and that's how they look like, basically. So basically, a hydrogel with micro patterns, and inside the micro patterns we have two different cell types, and inside the hydrogel we have dermal fibroblasts. And when we want to do the vascularization, we add also endothelial cells together with the dermal fibroblasts. So we have at least 4 different cell types to start with in a physiologically relevant extracellular matrix.
Are you just trying to grow the hair follicles amidst the soup of cell types, or do you have a piece of skin and then you're trying to grow a hair follicle on the edge of it or in the center of it, let's say?
You mean growing these growing hair on a piece of skin that's harvested?
On an existing piece of skin, yeah. Maybe that would tell you something different. Even if you had like a small skin island and you try to grow hair in it or through it or on the edge of it. It's just an idea for a different type of construct, instead of a soup of cells.
Yeah, I mean, I wouldn't be surprised if we collected skin, let's say mouse skin or human skin, and do a punch biopsy, or like make a hole in the middle of it, and then seed our constructs or seed our cells in there within this hydrogels, with the signaling coming from the surrounding tissue that you collected. I wouldn't be surprised that would enhance this process because of the signaling that we just talked about previously. You're basically isolating some of that microenvironment, and then doing your experiment, although incomplete, still in a natural environment, and more natural than what you have in petri dishes or plastic culture dishes.
Well, it sounds like you want to find out the minimum required to grow hair follicles. So, again, if you started out with a real small piece of skin with, 1 or 2 hairs in it, maybe that's enough. And that would help you grow a whole bunch more. And then you'd be off to the races. I don't know. So it just seems like it would be, the mouse model works. On regular people, it works with all the factors. And then you're trying your soup of X number of cells, and that's kind of working. So maybe pick a few intermediate steps and try to interpolate so you can get closer to what you need faster. Just an idea for you,
Well,
maybe I'm simplifying it. It's much harder to do that than it sounds. I don't know.
I'm not sure if I completely understood your suggestion.
That's what I was asking. Does the construct have any shape to it, or is it just cells, in a medium that are free floating, and they're all just sitting there in a small area, and from there a follicle forms? Or do the cells have structure when you're trying to grow your hair follicle? What does the construct look like? Maybe a little bit of detail besides just the cells that are involved. Like, what does it actually look like? Does it have structure when it starts out?
Yeah. Thanks for clarifying that, because I didn't really talk about the geometry and how it looks actually. It's hard to tell, without pictures. But so we basically have a transwell. We call it a transwell culture system. So it is a transwell. It's like an insert that we put in a plastic dish. It's a circular well, and then it has a membrane attached to it at the bottom. So and then this is suspended, in a culture dish, And in the circular transwell, we add our hydrogel, which is a liquid form first. And in this liquid form, we have the dermal fibroblast, and it gels first in a circular shape, taking the shape of our transwell. It's around 2 centimeters in diameter. And once it gels, once it's solid, actually before it gets solid, we put our 3D printed molds. Those are plastic molds that have the hair follicle-like extensions on them. We put them on top, and they float, these molds. So, and they have the hair follicle extensions. And once this hydrogel forms around these hair follicle-like extensions, we can peel off the mold. So we get rid of the mold from top, and now we are left with open channels on top. So we are left with open holes that look like hair follicles, that have the geometry of the hair follicles. For example, these holes are around 300 microns in diameter, and they're around 4 millimeters in length. And we could control these parameters with 3D printing, and we could control the hair density, for example. We can make different shapes of, different patterns of hair follicles with this technique. So it's basically a solid circular shape, around 2 centimeter diameter in size. It constructs, and then you add your cells into those holes, and then they start communicating with each other, and then they grow more. For example, you may start with a 4 millimeter, in length of microchannels, but once the cells, start differentiating, they can grow like they do in hair morphogenesis during, embryonic development. They grow down into the dermis. So in this case, they're growing down into our constructs, into the hydrogel. So they're elongated, starting from 4 millimeters, but then they become much longer, like they do in the body, and they can have an angle also. For example, we start with a 90-degree angle. These holes are perpendicular to the surface of the gel, but then once these cells start, interacting with each other and start their differentiation and hair growth, they can change the angle. They can become they can pick up a more physiologically relevant angle. So what we found in our constructs, actually, after a week or so, we have around 45 degrees of an angle of these hair follicles when we image them in 3D using 3D imaging, confocal imaging. We saw that these hairs now have an angle that is mimicking the physiological angles of the hair follicles, which this was to our surprise. We started with 90 degrees, and then they somehow find the right angle when they start forming.
Yeah, that's really interesting. Makes me think of plants, that go towards the sun. I wonder why hairs will go from 90 to a 45 degree, and in which direction, and if they change direction. That's very interesting.
Yeah.
Huh. Okay.
And we can, with these models now, since now we have all these in our hands, we can start asking these questions and answering some of them, that we weren't able to answer before, because in humans, we don't have much knowledge about human skin. What we've done so far, we know so much about mouse skin, for example, or rodents or other animals, but we don't know about human skin that much, human hair follicles that much, and how these things form, during embryonic development, for example. Now we can look into, we can monitor what happens in the early stages of hair follicle development.
So what's your end goal? What would be a great result for you, in the near term, in the next few years, for you to figure out?
Well, first of all, I would love to see, love to see how we can grow hair follicles in vitro completely without having to put it or grafting it onto mouse. So finding the right formulation to have in our culture medium that would, enhance this efficiency of hair growth, human hair growth in vitro. So that would be my goal, let's say, for the next 2 years, which we're working on currently. I mean, we have many different projects going on in our department and in my group, but this is the most relevant one to this topic, and I would love to see that happening. And also another thing that we're looking at is now, as I told you before, hair follicle is not only composed of a hair shaft and 8 layers of epidermal cells and dermal papilla cells. You have the other compartments. For example, there is the stem cell compartment. There is the sebaceous gland compartment, which is the fat factory of the hair follicle. So adding these components to hair follicle will make it a fully formed hair follicle. It's a physiological hair follicle. So I want to add all these components. For example, there is also pigmentation, right? We don't now the hair that we grow is unpigmented, so it's white hair basically, because it doesn't have it doesn't have pigment. It doesn't have the pigment making cells, which are melanocytes. So these melanocytes are located at the hair matrix above the dermal papilla cells and inside the epidermal compartment of the hair follicle. But in this study, we didn't incorporate any melanocytes into those channels. But it's possible. It's possible to layer all these cells on top of each other in the formation that is in the body. So that's one of the goals also. Now we want to add these melanocytes and get pigmentation and grow colored hair.
Well, for listeners, what's the best way for them to learn more? Maybe see pictures of what you're working on. Read papers. What's the best way for people to find out more?
Well, they can check out our new paper in Nature Communications. It got published late last year, December 2018. And they can also check out the video that is on Columbia University's website. So Angela Cristiano, she was the corresponding author of this paper, and she talks about the study, the details, and then I'm also on that video talking about the engineering part of this study, and they can learn much more about the study on that video, which was shared with the rest of the media. Actually, it came up on in different countries, also in the media, sometimes with very exaggerated titles, which I'm not very happy with, like cure for baldness, the end of baldness. So those are unfortunately not very accurate titles.
Well, very good. Well, Erbil, thanks for coming on the podcast. I appreciate it.
Yeah, thank you so much, Richard, for having me. It was really nice to talk about with you.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.